everything we've seen so far, how does a hardworking cell do it all? Every single cell has got to be able to carry out multiple catabolic and anabolic pathways at once. With so much going on simultaneously, how do cells keep track of what needs to happen? What mechanisms direct the flow of biochemical traffic? And how do cells ever get anything done if one set of reactions reverses what is happening in another? In multicellular organisms, metabolic control is even more complicated because specialized cells in different parts of the organism must work in harmony. In this lecture, I will focus primarily on controls inside each individual cell and the simple means cells use to manage complex and interconnected metabolic pathways. We'll also look at a macro cycle between muscles and liver and touch on how control activities are fine-tuned in response to changing internal conditions. We'll trace how cells respond to exercise as well as to a sedentary lifestyle by adjusting what happens in metabolic pathways. You'll also discover the secret of so-called warm-blooded animals, what newborn babies and hibernating grizzly bears have in common, and how researchers hope to apply this knowledge to combat obesity. And you're going to learn about a drug from the 1930s that helped people to burn fat in their sleep as it killed them. Now, a remarkable thing about metabolic control is that there's no central processor in a cell that directs activities through any sort of master plan. Instead, their responses to varying conditions are built into the enzymes that each run their own small piece of the show. There's no cellular brain that keeps tabs on everything that's happening and dials up all the pathways up or down from a central location. But this is not as surprising as it seems at first glance. How a cellular pathway is controlled is often no more complicated than the humble flush toilet. This device, as we know, automatically refills its tank when it's emptied, but stops the inflow of water when it reaches a certain level. Like the toilet, many cellular mechanisms operate through simple if-then responses. If a certain condition is met, then a particular response automatically follows. So that word control should be put into quotation marks because it refers to the built-in responses of cells and tissues to very specific conditions. Now, with that caveat in mind, let's talk about control. We've already learned ways that enzymes can be controlled. These include allosteric regulation, where the binding of a molecule modified an enzyme's activity. There's covalent modification, where a phosphate group gets chemically linked to specific side chains in an enzyme protein to turn it on or off. There's also control of whether or not a cell even makes an enzyme in the first place. In fact, the ability of enzymes to be regulated by a variety of means is very important. Enzyme activity can be tuned to increase or decrease in response to a variety of conditions. This property is used to great advantage in controlling metabolic pathways. We'll first take a look at how regulation of key enzymes in certain pathways ensures that a given cell doesn't waste energy breaking down and building the same molecules simultaneously. And since neither the synthesis nor the breakdown process is 100% efficient, it will take more energy to build the molecules than is obtained by breaking them down. In breaking down glucose and glycolysis, for example, we saw that the net products were 2 pyruvates, 2 NADHs, and 2 ATPs. To make glucose in gluconeogenesis requires 2 pyruvates, 2 NADHs, 4 ATPs, and 2 GTPs. Put glycolysis and gluconeogenesis together and you see that there's a net loss of 4 triphosphates each time you run both cycles. This is an example of what biochemists call a futile cycle. It's futile because metabolically it's pointless. In fact, there's an energy loss accompanying it. Now, that is not a prescription for success. This is a cellular equivalent of spending money faster than you make it. Cells, unlike humans, don't have overdraft protection, so if a cell kept running both pathways at once, it would not be very long before it ran completely out of energy and died. Naturally, it's in a cell's interest to prevent this from happening. So let's take a look at how this works for glycolysis and gluconeogenesis. Now, thinking back to glycolysis, the most important regulatory enzyme for breaking down glucose is PFK at step number 3. You may also remember that gluconeogenesis didn't try to reverse the PFK reaction. Instead, it employed a different enzyme called fructose 1,6-bisphosphatase, or F16BPase. These two key enzymes control the flow of intermediates through glycolysis and gluconeogenesis, respectively. A simple way to ensure that the two pathways don't run simultaneously would be to turn off one of the enzymes at any time the other one is turned on. And this is exactly what the cell does. These two enzymes are regulated such that both cannot be active at the same time. The device that controls this is impressively simple. A switch that turns one enzyme on is also a switch that turns the other off. How? Well, both enzymes are allosterically regulated by the same molecules, but in opposite ways. So for example, AMP is a regulator that activates the glycolysis enzyme PFK, but it inactivates the gluconeogenesis enzyme F16BPase. AMP stimulating glycolysis makes metabolic sense, as AMP levels are high when cells are low on ATP. More glycolysis would provide more ATP, while gluconeogenesis would use it up. A more important regulatory switch with the same results is fructose 2,6-bisphosphate, or F26BP. This is another molecule made in cells that activates PFK and glycolysis to eat up all the glucose, while it inhibits the glucose-making F16BPase and gluconeogenesis. By contrast, a regulator where both effects are reversed is citrate. And where is citrate formed? Well, you remember, in the citric acid cycle. Citrate accumulates when cells have abundant energy. When lots of citrate is present, glucose breakdown is going to taper off to slow down ATP formation. On the other hand, high levels of citrate activate fructose 1,6-bisphosphatase, which will cause more glucose to be made, thus using ATP to store energy. Regulation is simple when a single molecule has opposite effects on regulatory enzymes of corresponding catabolic and anabolic pathways. Such regulation is called reciprocal regulation. It's convenient and especially important when the corresponding catabolic and anabolic processes can occur in the same cellular location. In this case, both sets of reactions are happening in the cytoplasm. Reciprocal regulation also plays a role in the building and breaking down of glycogen. Glycogen, let's recall, is made when cells have more glucose than they can immediately use, and is broken down to release glucose when energy is needed. The regulated enzyme for glycogen breakdown is called glycogen phosphorylase, and the corresponding enzyme for glycogen synthesis is glycogen synthase. Both enzymes are regulated by phosphorylation and dephosphorylation. Again, the switch that turns one enzyme on turns the other enzyme off. Phosphorylation makes glycogen phosphorylase more active and glycogen synthase less active. Of course, the results reverse when the phosphates are removed. Less glycogen breakdown, more glycogen synthesis. The stimuli for phosphorylation, dephosphorylation of these enzymes is hormones that mount a cellular response to bodily needs. Epinephrine, also known as adrenaline, is released in times of stress or excitement. It acts to stimulate a cascade of phosphorylation, which activates glycogen phosphorylase. This favors the release of glucose from glycogen to fuel muscles and prepare them for action. The hormone insulin, on the other hand, is produced when blood levels of glucose increase. Insulin stimulates dephosphorylation, activating glycogen synthase and favoring storage of glucose as glycogen. So we've seen two methods for control of key enzymes in major metabolic pathways: allosteric effectors in glucose metabolism and phosphorylation in glycogen metabolism. In both, the on switch for one pathway is the off switch for the other, thus reducing the chance pathways for breakdown and building of a molecule operate simultaneously in the cell. A third important mechanism for regulating metabolic pathways is the availability of substrates, specifically electron carriers that are needed to run the reactions. Now, you're already familiar with electron carriers from the lecture about electron transport. The electron carriers NAD+ and FAD accept electrons released in cellular oxidation reactions to become NADH and FADH2, which are the fully loaded electron carriers. The relative availability of oxidized electron carriers like NAD+ compared to reduced carriers like NADH is a factor in metabolic control. Other players include the proton gradient made by those electron carriers, but also ADP and oxygen. To understand the phenomenon of metabolic control, let's first review what happens in electron transport. Movement of electrons through the electron transport chain pumps protons out of the mitochondrial matrix, creating a proton gradient. The terminal acceptor of electrons during electron transport is molecular oxygen. The proton gradient generated by electron transport is used by ATP synthase to create ATP from ADP and phosphate. All of these components are interdependent on each other. If one is limiting, all are affected. Now let's imagine a sedentary person sitting, eating pizza, drinking beer, and watching TV. I'll call him or her Sam. Sam isn't moving much, except to occasionally push buttons on the remote. With plenty of food available and no exercise, ATP levels in Sam's cells are high. ADP levels are low, having been mostly used up to make ATP. When this occurs, the ATP synthase slows to a crawl. ATP synthase is the cellular enzyme through which the protons re-enter the mitochondrial matrix in the process of oxidative phosphorylation. If ATP synthase slows down, the proton gradient grows, so long as the electron transport system is running. So for a while, it's like a bathtub with a partial plug in the drain. If the faucet keeps running, the water level will slowly rise. But this can't go on indefinitely. Unlike a bathtub that can overflow, after a point, buildup of the proton gradient is no longer possible. It's more like a rechargeable battery nearing full capacity. The proton gradient, let's remember, depends on the movement of electrons through the electron transport complexes and vice versa. If the proton gradient gets fully charged, electrons can't move in electron transport. When electrons stop moving, oxygen use drops. That's why couch potatoes don't breathe heavily when they're sitting and eating. So Sam's sluggish electron transport system from lack of exercise means NADH and FADH2 have no place to hand off their electrons. The change of NADH back to NAD+ and FADH2 to FAD slows down. As NADH and FADH2 concentrations increase, NAD+ and FAD concentrations fall. And when this happens, oxidative pathways relying on NAD+ and FAD by necessity decrease too. This means that the citric acid cycle and fatty acid oxidation slow or stop. Lower citric acid cycle activity and decreased fatty acid oxidation explain what we know from observation. If you don't exercise, you're not going to burn off fat. Sam's whole system got plugged up because there wasn't any exercise to convert ATP to ADP. Let's look at what happens when ATP starts getting used. This might happen as a new year rolls around and Sam decides it's time to get moving and drop some pounds. Sam starts to jog. The muscles in Sam's legs use ATP to fuel their movement, leading to a drop in ATP levels. The lower ATP levels from running have another effect. ADP levels increase, so Sam's ATP synthase begins to spin a little faster. As a consequence, more protons begin to enter the mitochondrial matrix. The proton gradient decreases. With a smaller proton gradient, electron transport speeds up. And when this happens, cells need more oxygen to give the electrons to. SAM begins to breathe more heavily to supply the needed oxygen via the bloodstream. And as more and more electrons flow, more NADH and FADH2 get converted to NAD+ and FAD. Increased levels of these carriers stimulate the citric acid cycle and fatty acid oxidation. And as more fatty acids get used up, SAM begins to burn off those extra pounds. By exercising, SAM unplugged the system. What SAM's experiences show is the codependence of factors in electron transport, oxidative phosphorylation, and oxidative metabolism, such as occurs in glycolysis, fatty acid oxidation, and the citric acid cycle. If cells had unlimited quantities of NAD+, FAD, and ADP, then stopping electron transport or oxidative phosphorylation would have no upstream effect on the citric acid cycle and fatty acid oxidation. But electron carriers and ADP are limiting resources. They must be recycled by electron transport and oxidative phosphorylation. In short, oxidative metabolism, electron transport, and oxidative phosphorylation all respond to changes in each other. This is what metabolic control is all about. Now, there's one more interesting thing that can happen if SAM decides to run really hard. When people exercise vigorously, their bloodstream has difficulty supplying all the oxygen the muscles could optimally use to generate ATP. What happens when cells have insufficient oxygen? Well, electron transport slows. NAD+ levels fall, and that could be a real problem if you're exercising heavily. Now, fortunately, there's a backup. Fermentation, we've heard about that before. In fermentation, oxygen is not needed, and cells convert pyruvate to lactic acid to regenerate NAD+. That would normally require oxygen, but thanks to fermentation, we get NAD+. When SAM is running really hard, lactic fermentation serves as an alternate source of NAD+ to keep glycolysis going. Cells need glycolysis at a minimum because it can provide ATP even when the citric acid cycle and fatty acid oxidation are stopped. But it takes almost 20 times as much glucose to make ATP in fermentation as it does in the presence of oxygen. So SAM's muscles need a lot of glucose to keep going. Now, fortunately, SAM's liver is up to the task, assuming it wasn't damaged from too much of the beer drinking earlier. As levels of glucose in the blood fall, the liver releases glucose into the bloodstream using both gluconeogenesis and glycogen breakdown as quick and easy sources of glucose. That's cool. Meanwhile, back at the muscle cells, fermentation is churning out lactic acid. Lactic acid is a metabolic dead end. It's made from pyruvate, but the only thing lactic acid can be metabolized to is pyruvate again. Reoxidation of lactic acid back to pyruvate requires NAD+, but that's in short supply in the muscle cells. What do? Well, rather than accumulate lactic acid that's useless and even unhealthy if enough is on hand to lower the pH, the muscle cells instead dump it into the bloodstream. And the lactic acid travels to, you guessed it, SAM's liver. Now, unlike the muscle cells, the liver is not exercising and has no shortage of oxygen from its neighbors, the lungs. As a result, the liver has electron transport going, so it has plenty of NAD+. The liver cells use this NAD+ to convert lactate back to pyruvate. Now think about where this is going. Lactate is not a dead end for the liver, from which it makes pyruvate, and pyruvate is then used to build glucose through gluconeogenesis. So the liver cranks out glucose and dumps it into the blood for muscle cells. Now the circle is closed. Muscle cells export lactate to liver cells, which convert lactate to glucose, which muscle cells take up and use to make more lactate. This macro cycle of metabolites traveling between the muscle cells and the liver is called the Cori cycle. It was named for Carl and Gertie Cori, the husband and wife team who worked out the connections between liver, glycogen, hormones, and blood glucose to win a Nobel Prize in 1947. Now let's compare the energy used when the Cori cycle is running to when it's not. When people exercise vigorously, they breathe very heavily as their bodies try to provide enough oxygen to their cells to keep electron transport going. There are two possibilities. First, that breathing provides enough oxygen so that electron transport can work efficiently. This is the aerobic scenario. In the second situation, the individual is exercising even more rapidly than oxygen can be delivered, so the Cori cycle is going full force. This scenario is partially anaerobic. In both situations, Sam is doing what we would in everyday speech describe as aerobic exercise. The exerciser is breathing hard with an elevated heart rate. In the aerobic, well-oxygenated scenario, Sam gets approximately 36 ATPs for each glucose completely oxidized. In the anaerobic scenario, Sam runs gluconeogenesis in the liver while the muscles run glycolysis plus fermentation. The liver then expends 6 ATPs to make each glucose that the muscles only obtain 2 ATPs from in fermentation. In the well-oxygenated scenario, there's no cost of using the liver to supply muscles with glucose, so all of glucose's energy is available to the muscle cells. In the anaerobic scenario, the cost is 6 ATPs in the liver to supply glucose worth 2 ATPs in the muscles. Yeah, that's a high price to pay, but if the body doesn't pay it, the muscles don't work. You can do the math here. Since muscles in the oxygenated scenario deliver 36 ATPs from each glucose, and muscles running fermentation get 2 each, muscles in fermentation need 18 times as much glucose to get the same energy as muscles rich in oxygen. One of the important advantages of metabolic regulation is to prevent cells from running futile cycles. Carrying out glycolysis to break down glucose while simultaneously synthesizing glucose within the same cell is wasteful and could ultimately be disastrous. And yet some cells do break free of metabolic regulation and run a cycle that is perversely inefficient, almost like a one-sided futile cycle. Why would cells do that, you might wonder? Well, wonder no more. There's metabolic method in this madness. It is a basic fact about chemical reactions that some of their energy is lost as heat. Warm-blooded animals like us eat a lot more food and have a lot more mitochondria in our cells to create and manage the heat that keeps us warm. Speeding up metabolism is a great way to generate heat, but how does one do that? Well, in us, the hypothalamus and liver play important roles. A futile cycle might be encouraged as one source of heat. In any case, the liver does a great deal of metabolism, so heat generated there gets passed to the blood that disperses it through the body. The hypothalamus monitors this temperature and uses hormones where necessary to stimulate metabolic processes. Cold-blooded animals don't eat as much as we do, and they don't have as much in the way of energy resources. And last, they don't monitor, so they're pretty much stuck with the temperature of their environment. Human infants run a type of futile cycle in specialized fat cells known as brown fat. Brown fat gets its name from the fact that these cells actually have a brownish color due to an abundance of mitochondria. Brown fat is also populated densely with capillaries for providing nutrients to the cells and distributing heat produced by them throughout the body. In brown fat, electron transport occurs in the numerous mitochondria, just as it does in other cells. In most cells, as you know, electron transport results in the buildup of protons in the intermembrane space, and the proton gradient across the membrane powers ATP synthesis. In brown fat, though, the concentration of protons in the intermembrane space doesn't go up. And this is because the mitochondria of brown fat have a protein called uncoupling protein. Uncoupling protein does something weird. It makes the mitochondrial inner membrane permeable to protons. So when protons get pumped into the intermembrane space, they leak right back into the mitochondrion. With no proton buildup, there's no synthesis of ATP. What happens to the energy that is not harvested as ATP? It's released as heat. We call this phenomenon uncoupling because electron transport is no longer linked to the production of ATP. Now, normally when cells have plenty of ATP, ATP synthase slows down. But protons can normally cross the membrane only through the ATP synthase, so they pile up in the intermembrane space. And when protons pile up, the battery gets fully charged and electron transport slows. But when protons leak across the membrane, the proton concentration in the intermembrane space never goes up. The battery never gets charged. So electrons continue to flow because there's no buildup of protons to slow them down. Meanwhile, the cell is using ATP while making very little or none. With electron transport operating furiously, NADH and FADH2 are continuously being converted to NAD plus and FAD. And you may remember from earlier, abundant NAD plus and FAD fuels the citric acid cycle and fatty acid oxidation. So these cycles run much more than they would otherwise, just as if you were exercising. And when that happens, heat is generated, just as it is when you're working out intensely. Now, it is thought that brown fat is abundant in infants because they're not yet good at thermal regulation. The solution, of course, then is to run wasteful pathways that burn abundant baby fat and use the heat that it generates to warm the body. Using uncoupling proteins to generate heat isn't unique to humans. Hibernating animals, from grizzly bears to ground squirrels, use the same strategy to periodically raise their body temperature during their long winter's nap. Burning fat during sleep, pretty cool idea, huh? Babies and bears have hit on a trick that many of us would like to use. They burn fat without so much as wiggling a toe. Many of us would like to replicate this feat, could we? Well, adult humans, as it turns out, do have some brown fat. Interestingly, skinny people seem to have more, suggesting brown fat may play some role in metabolic regulation. Increasing the proportion of brown fat cells with their greater fat-burning ability could help in treating obesity. Now, in the 1930s, one attempt to replicate what happens in brown fat used a diet drug that mimicked the effect of uncoupling protein. It's called 2,4-DNP, or simply DNP, and the drug was able to permeabilize the membranes of mitochondria to protons, just like uncoupling protein. 2,4-DNP causes cells to speed up metabolism just like uncoupling protein. It was very effective at causing weight loss. Where uncoupling protein is only in brown fat, 2,4-DNP could function in all cells. But that unfortunately turned out to be too much of a good thing. Remember that uncoupling protein is useful for generating heat. When that happens in a small subset of your cells, you become slightly warmer, just as a fire in the fireplace raises the temperature of the room by a few degrees. Taking DNP, by contrast, is like setting fire to your whole house. 2,4-DNP killed many people and caused serious harm to others, and their deaths were from hyperthermia, with the fatal body temperatures reaching up to 110 degrees. Makes hopping on a treadmill sound pretty good, doesn't it? An ode to DNP. I saw my friend the other day. His face had changed. His hair was gray. I saw his hand. It seemed quite warm. I asked him, could he please inform just what had happened to him since he'd gained some heated fingerprints. He looked at me and with a shrug said something about a diet drug that promised him a radiance, adjusting proton gradients. I said, oh no, don't say to me that you've been using DNP. It's true, he said, that stuff I ate in hopes that I would lose some weight. A week went by and once again I ran into my mixed up friend. I asked him how he felt that day. He smiled at me a funny way. Said, I stopped that DNP I bought and now I'm not feeling so hot. Current research has given up on the idea of mimicking uncoupling hormone. New approaches involve attempts to activate and increase numbers of existing brown fat cells. One approach comes from a drug called mirabegron, which blocks hormone receptors on existing brown fat cells. This stimulates them to take in glucose and other nutrients and increase metabolism to produce heat and energy. Yet another lead is a protein hormone called irisin that appears to increase brown fat stores. And what does irisin do? Well, in fat cells, it may be increasing production of uncoupling protein, the very thing that helps burn off fat. But here's one interesting tidbit. Irisin is also known as the exercise hormone because its levels are increased by exercise. So if you want the effect of losing fat in your sleep, it seems the thing to do is exercise when you're awake. It looks as if there's no getting around physical activity as a way to burn fat. I think of the butterfly effect whenever I think about metabolic control. This is the idea that the fluttering of a butterfly's wings in one part of the world ultimately affects weather on the other side of the globe. What it implies is an interconnectedness and interdependence such that every manipulation of the system in one part has an effect elsewhere. Considered in isolation, a gradient of protons might seem unrelated to the spinning of the citric acid cycle. But what we've seen is that they're closely connected. Butterflies may not cause hurricanes, but the fluttering of electrons in mitochondria really does control a cell's metabolic weather.